Hi there, science fans. Welcome to another episode of Science with Dr. Dan. This channel brings you recent advances in science, especially scientific achievements relating to current and future medical treatments. In these episodes, I try to explain these achievements in terms that I hope are understandable to my audience. If some of you would like a more in-depth explanation of the subject I am presenting, please make a request in the comments section. The present episode is entitled Child's Cancer Cured Using Cellular Therapy. I describe how a 13-year-old girl suffering from an aggressive blood cancer has been cancer-free for almost 2 years. The clinical trial was evaluating a new technology called base editing. If you like this video, please click the like button and please subscribe to this channel and click the bell to be notified of new videos. A 13-year-old girl named Alyssa was diagnosed in May of 2021 with T-cell acute lymphoblastic leukemia, or ALL. Leukemia is a type of cancer that affects the blood and bone marrow. It is characterized by the uncontrolled production of abnormal white blood cells, which can interfere with the normal functioning of the blood and the immune system. One type of leukemia is called acute lymphoblastic leukemia, which affects a class of white blood cells known as lymphocytes. ALL is a common form of childhood cancer, and it can also affect adolescents and adults. However, children and young people are the most commonly affected group. The exact cause of ALL is unknown, but certain factors such as genetic mutations and exposure to radiation and chemicals may increase the risk of developing the condition. The good news is that with early diagnosis and proper treatment, the cure rates for children with ALL have significantly improved in recent years. The current cure rate for children with ALL is approximately 80 to 85%, and for adults is around 65 to 70%. The success rate of treatment is dependent on several factors, including age, type of ALL, and the response to treatment. The primary treatment for ALL is chemotherapy, which uses drugs to destroy the abnormal cells and stop them from spreading. Chemotherapy is usually given in several cycles, with each cycle lasting several weeks. The main problem with the use of chemotherapy is that it is nonspecific. It doesn't discriminate between normal and abnormal lymphocytes, thus destroying both normal and abnormal tissue. Other treatments may be used in combination with chemotherapy, including radiation, stem cell transplantation, and targeted therapies. In the case with Alyssa, her cancer was aggressive, and all treatments had failed. So she was placed in an experimental human clinical trial using DNA-based editing technology. So what is base editing? Base editing is a method of gene editing that allows the precise correction of specific mutations within DNA without introducing double-strand breaks. Base editing permanently changes a single letter in a patient's genetic code at a specific location in the gene. This change results in silencing, repairing, or modulating the expression of that gene. This is an extremely powerful gene engineering tool that can reverse genetically inherited diseases, as well as train one's own immune system to track down and eliminate cancer cells without affecting the patient's normal cells. As can be seen in this slide, current base editors can only change a cytosine, or C, to a thymine, or T, or an adenine, or an A, to a guanine, or G. Present DNA editing is a complex molecular mechanism that uses several genetic engineering tools to convert one nitrogenous base in the DNA to another. If you would like to see an expanded video describing these molecular conversions, please leave me a comment and I will make such a video. In order to have a clearer picture of base editing, we'll briefly review the structure and function of the DNA molecule. DNA, or deoxyribonucleic acid, is a molecule that contains all of the genetic information needed to produce, maintain, and grow a living organism. DNA is the building block of life, and its structure and function is crucial in understanding the biology of all living things. Panel A shows the structure of DNA. DNA is composed of 4 types of nitrogenous bases: adenine, thymine, cytosine, and guanine. These bases pair up to form the rungs of a ladder-like structure known as a DNA double helix. The rungs are held together by hydrogen bonds between the bases, with adenine always pairing with thymine and cytosine always pairing with guanine. The 2 strands are complementary to each other and can be thought of as a code, with each base representing a letter in the code. The sequence of bases determines the genetic information within the DNA molecule. The code is read by other cellular machinery and translated into proteins, which are the building blocks of cells that carry out a variety of functions. If you would like a more in-depth presentation of the structure and function of DNA, please leave me a comment in the comment section. Panel B is a cartoon that represents a section of the DNA containing a mutation at a single base cytosine. That is pictured in the lowercase a part of panel B. This particular single base mutation renders the cell as abnormal. The complex base editing molecular machinery recognizes the mutation at the letter C and converts that C or cytosine to a Th or thymine. This conversion takes place by a precise scissors-like cutting mechanism. The conversion of the C to T results in the formation of a cell with the desired properties. This slide indicates how the treatment given to Alyssa works. Lymphocytes are a type of white blood cell that plays a crucial role in the immune system. They help to identify and eliminate pathogens such as viruses, bacteria, and other harmful invaders that can cause illness and disease. There are two types of lymphocytes, T cells, which are also known as T lymphocytes, and B cells that are also known as B lymphocytes. T cells help to directly kill infected cells, while B cells produce antibodies that help neutralize the pathogens. Step 1 in the panel depicts a cartoon of the majority leukemic T cells in Alyssa's body. These cells have a variety of surface membrane molecules that allows them to escape detection from Alyssa's immune system. In order to develop a cellular therapy to train immune cells to attack these leukemic cells, her doctors needed to obtain normal T cells from a donor and edit those donor T cells in 3 ways. In step 2, the first base edit disabled the T cells' targeting mechanisms so they would not attack Alyssa's body. The second base edit removed the membrane surface marker called CD7, which is on all T cells. The third edit provided an invisibility cloak that prevented the donor cells from being killed by a chemotherapy drug. The final stage of the genetic modification instructed the donor T cells to go hunting for anything with a CD7 membrane surface marker on it so that it would destroy every T cell in her body, including the cancerous ones. That is why the donor CD7 marker had to be removed, otherwise it would have destroyed itself. After the success of the treatment, Alyssa's immune system, including T cells, were reconstituted with a bone marrow transplant, and she has been cancer-free for the last 2.5 years. Around 60% of human genetic diseases are the result of a point mutation, or a single base alteration in a gene. Base editing technology is a tool that is capable of point mutation correction. The work of David Liu and his colleagues was published in 2016 and indicated that this molecular tool can be used to treat individuals suffering from inherited diseases. In a short 5 years, this technology was brought from the lab to human clinical trials. This slide indicates some of the clinical trials presently underway and includes acute lymphoblastic leukemia, familial hypercholesterolemia, sickle cell disease, beta thalassemia, and some blood cancers. While base editing is still in its early stages, the results so far are promising, and researchers are optimistic that it will play a major role in personalized medicine. Thank you for watching, and if you like this video, be sure to click the like button. Please subscribe and click the bell to be informed of future videos. Also, if there are specific topics you would like me to research and present, be sure to comment in the comment section. Bye.